So thank you, Colin, for the very kind words of introduction. Is this a good volume? Wonderful. Okay, so what I will try to do is take you through a bit of a story this evening that hopefully will be finished, I think you said by about 9, yes? Yeah? Good. Just making sure everyone's awake. No, a bit of a story that encapsulates some of the work that my lab has done and shifts it into a bit of an aging context. Okay? However, before we start addressing the overall story, what I want to do is just put a few pieces of important items out onto the table. And the first of those is going to be discussing this idea of successful aging. So by way of introducing that point, what I would want you to do is just imagine for a moment the amount of time that it might take you to run 100 meters. So about how long do you think it might take you if you were very, very motivated? Let's imagine there's a nice, large, double cappuccino latte with an extra vanilla squirt or whatever it is you happen to like at the end of that 100 meters. How long would it take you? 9.58 seconds, okay? It's very precise. Anyone else? Yes? 12 seconds. Okay. 9.58 seconds would be a wonderful time. In fact, it's more or less the official world record set by Usain Bolt. What I want to do, though, is in the context of the discussion, introduce you to Hidekichi Miyazaki. So Mr. Miyazaki ran 100 meters in 42.22 seconds at the age of 105. Now this, believe it or not, wasn't his personal best. He set that a few years prior to this point, 34.10 seconds, at the very ripe age of 103. Now, unfortunately, Mr. Miyazaki died earlier this year of a cerebral hemorrhage, nearly 110 years of age. The reason why I introduce him in particular is that he's an exemplar of this notion of successful aging. And if you start to go through the literature, what you find is that there's a lot of discussion and debate over exactly what it is we mean by successful aging. However, a good template or a good framework to discuss the notion was articulated about 30 years ago by Rowe and Kahn. And they outlined 3 core principles of successful aging. One of these is that as individuals age, they retain fairly good, fairly reasonable physiological capacity. So, for instance, the ability to run 100 meters. They also see relatively low levels of disease and maintain good engagement socially and with life more broadly. So the first thing I want to introduce to you is this notion of successful aging, right, as epitomized from the biological side of the discussion by limited expression of disease and relatively good maintenance of physiological function. Good? Okay. So the next part of the introduction that I wish to sort of go through takes us on a bit of a time travel exercise. And what I want to do here is transport you to June 1944. And I want to take you to Northern Europe. And in the early weeks, the early days indeed, of June 1944, there was a dramatic event occurring on the northwestern beaches of France. And this, of course, was what? D-Day, right? The Normandy invasion. So there were hundreds of thousands of Allied troops that moved into this part of France and began to push northward through France, through Belgium. And over the course of the ensuing couple of months, they pushed forward, moving German forces back as they went. Unfortunately, in early fall, their advance was stopped as they moved partway through the Netherlands. Right around that time, the Dutch resistance started trying to contribute to the Allied efforts by calling for a general rail strike. Unfortunately, the rail strike, the Allied advance, just wasn't enough, and Nazi Germany decided to punish those areas of the Netherlands still under their control because of the resistance efforts. And as a result, the areas of the Netherlands that remained under Nazi Germany's control had a very strict food embargo placed onto them. And this food embargo, together with a very vicious winter cycle, led to a very quick and dramatic drop in the daily caloric intake of people in that part of the country. In fact, intake went from 16 to 1800 calories down to between 400 and 600 calories. And just to give you a point of reference, perhaps you've gone to Tim Hortons at some point over these last few days, and maybe you had a small coffee, double-double of course, because we're in Canada, and a banana muffin. Well, together, that would give you about 600 calories. So imagine, if you will, going from a typical caloric intake to no more than what you would see in a coffee and a muffin. And this started in late fall 1944 and continued until spring 1945, when the Allies finally managed to rout the German army from that part of the Netherlands. Okay? As a result, in very quick fashion, caloric intake returned to typical. Now, what's interesting is a couple of years later, a number of pediatricians and clinicians started to take notice of some very interesting changes. Clement Smith from Harvard was probably most notable in this regard. And what he found was that individuals who gave birth during this time period gave birth to babies that were far below typical birth weight. And this sort of piqued a lot of interest. And over the ensuing few decades, there was examination of this birth cohort, individuals who had been born during or shortly after what's come to be known as the Dutch hunger winter. And what they found is a whole host of very interesting effects in terms of risk profiles for many of the common degenerative conditions that public health practitioners study. So, for instance, shifts in risk for things like obesity, blood pressure, and cancer. And indeed, this unfortunate natural experiment, together with a number of other similar events over the course of the 20th century, provided very interesting epidemiological evidence for the fact that the things that take place in utero in the very earliest days of our development, when organogenesis is taking place, when the foundation of the body is being laid down, that those events can put us onto a trajectory shifting risk for all different kinds of adult onset diseases. Now, what's interesting is that the vast majority of the work in this area has focused on metabolic diseases and peripheral diseases. Very little work has focused on diseases of brain. So you can see in the story of Mr. Miyazaki and the story of the Dutch hunger winter, the foundation for the story that I'm going to tell you now this evening. What my lab is in part interested in trying to understand is how events that take place in the very earliest times of our lives set the foundation for how our brains develop. Might it be that the maternal environment is influential in terms of whether or not our brains age successfully, whether or not we see an enhanced risk over the lifespan for different kinds of neurodegenerative diseases? Good? Excited? All right, so let's dive in. Now, all of the points that I've mentioned to this time have involved epidemiological studies and case studies and so on. My lab uses a lot of techniques from experimental physiology. So we're going to be shifting our focus down to the level of animal models. And we're going to start with the one that is predominantly used in this area of work, which is the rat. And over these last 30 or 40 years, there have been a number of studies that have tried to appreciate how it is diet-induced obesity affects our physiology by taking advantage of feeding animals different kinds of diets. So sometimes these diets are high in sugars of various sorts, sometimes fats of various sorts, sometimes fats and sugars. Right? And if you take a look at the literature, it's sort of very confusing to try to figure out exactly what the patterns are, because there's such diversity with regards to the experimental models and the outcome measures that are used. Right? But one thing that is typically the case is that people study male animals. And this has largely been because of financial reasons. Right? So you can imagine if you're existing on a limited research budget, you've got to spend that money in a very careful manner. And as a result, many people elected over the course of neuroscientific research history to invest those dollars looking at male animals. However, men and women do not share the same anatomy, the same physiology, the same endocrinology, the same developmental history. So it stands to reason that there are going to be differences, particularly in terms of how diet-induced diseases develop. Fortunately, over the last 20 or so years, there's been a gradual shift, and more researchers are starting to invest extra time and extra resources in understanding how diet-induced diseases develop uniquely in male and female rodents. And indeed, our group has been part of this shift. And that's the work that I'm going to talk about this evening. So what you can see here is a typical female Sprague-Dawley rat. And what we would have done for this particular experiment is place a group of animals either on a diet that sees 45% of their calories coming from saturated fats, or a controlled diet, a diet that's controlled, chemically defined in all the sorts of usual meaningful ways. Right? The animals were placed on the diets from the point at which they were young adults, before 5 weeks old for a typical lab rat, for a period of 10 weeks. And at the end of that, what we decided to do was a series of assessments at the level of organs, brain functional measures, and behavioral measures. Unfortunately, because I don't have until 9 o'clock, we won't be able to go through all of those, but rather we'll take a look at a few of them. And where I want to start is not really even at the level of brain, but at the level of trying to understand how energy-dense diets affect the overall structure of these animals. Right? Because you might remember from, say, health classes, if we want to study the problem of obesity in our species, we use something like a what? If you wanted to determine whether or not an individual was clinically obese, BMI, right? So you'd measure height, you'd measure weight, perhaps you'd measure waist circumference, something of this sort. Unfortunately, we can't really do that effectively in an animal. Right? This is one of the sort of technical complications, although in our species we have a reasonable understanding as to how to distinguish a normal weight from an obese human. There's no established criteria in animal models. It makes you wonder, because there have been hundreds and hundreds of papers, and yet no one in the field has actually established criteria. So one of the first things we wanted to do is actually figure out, well, what sorts of things change? And indeed, a lot of what had been done in male animals. And as I mentioned a moment ago, male and female animals, just like male and female humans, are not identical. So it's not enough to assume that what's true in one is going to be necessarily true in the other. So we set out to measure a whole series of biometrics to determine how it is diet-induced obesity develops in these animals. And I'll show you just some of those data. So on the bottom left, we have retroperitoneal fat pad weight and plasma leptin levels. Right? So we measured a whole series of different changes. So we looked at body weight. We did look at rodent BMI. So we measured length, nose to tail length, and waist circumference, all of these different sorts of biometrics. We didn't really see any change. Right? We measured food intake, caloric intake, and there was quite a bit of a difference. Right? Because rodents are not like us. Right? If you give a rodent a calorie-dense food, what you see is a shift in its eating pattern. Somehow, it actually seems to be aware that the food is calorie-dense, and it will eat less of it, as an attempt to sort of maintain its caloric intake. Right? If only we could do the same kind of thing, right? So we measured those kinds of eating patterns and so on. But in most of the measurements, we really didn't see much of a change. However, we did notice a shift in terms of retroperitoneal fat pads. So in rats, as in humans, there are different fat depots within the body. So post-euthanasia, we dissected those depots out, we measured them, we weighed them in different sorts of ways, and what we found is that the main lipid depot within the abdominal cavity is about 20% larger in female animals that have been on this calorie-dense diet. Seeing that, we decided now to go on and take a look at the blood samples we had collected and assess those for leptin. And if you sort of think back, if you've done a little bit of endocrinology or biochemistry, you know that leptin is critically important as a satiety factor and a metabolic regulator. And it's produced by adipocytes. So knowing that a high-fat diet led to an increased deposition of fat in these retroperitoneal fat pad depots, we had expected to see more leptin in the blood. And indeed, that's exactly what we found. So it's kind of nice, it's comforting when things begin to line up. The other thing that we assessed on the right is the ability of the animals to regulate sugars. And here we did the same thing, or very much the same thing as would be done in a clinic. Right? So if your clinician had concerns about your ability to handle sugar, what they would do is bring you into the clinic, give you a sugar solution, and then measure over time, usually 2 or 3 or 4 hours, the ability of your body to make use of that sugar. Right? In the case of the animals, what we do is we can't give them a cup of sugar solution, so we give them an injection of sugared solution, and then we do a blood draw for several hours. And then we use a glucosimeter to measure the amount of sugar in that blood. And we do this at various points, and what you can see, just examining the data in the bottom right, is that although the animals on that high-fat diet do seem to be gradually shifting, do seem to be gradually developing a problem handling sugar, it doesn't emerge to a point where it's a significant difference. Right? But again, keep in mind that these animals have been fed only for 10 weeks, and they were young, healthy animals to begin with. So quite likely if we had extended the amount of feeding, or had started the animals on these diets when they were older, or had given them a higher concentration of fat, we might have started to push the metabolic shift in a more dramatic fashion. Suffice it to say, though, that many of the changes that we see in our species with fat don't necessarily emerge in rodents, or at least not in the same sort of temporal pattern. So this is something to keep in mind. However, the data on the bottom left supported our idea that with these modified diets, we were able to bring about some kind of change in the animals. So far, so good? Right. So convincing ourselves that the diets were indeed able to shift the animals in some way, we now set out to see whether or not there was a change at the level of brain. And how might we do that was the question. Because if we had individuals in a clinic, we could do it reasonably well by simply having them come in and fill out different kinds of forms or participate in different sorts of neuropsych evaluations. Unfortunately, we can't do that with rats. However, about 3.5 decades ago, there was a technique that was developed that allowed us to assess episodic memory in rodents. And of course, episodic memory in rodents and in us is a reflection of the what, the when, and the where of our lives. So how do we assess that in rodents? Well, the fellow who managed to provide us with the technique worked at the University of Edinburgh at the time, and he was trying to figure out how to assess spatial memory in rodents. And the problem that he was presented with is that rodents have an enhanced sense of smell. So a lot of land-based mazes, as would have been typical at the time, really weren't appropriate because the animals could solve them and you weren't quite sure if they were actually solving the task or they were simply using odor cues from previous trials. And this is a perfect illustration of how sometimes simple twists of fate are so important. Because it just so happened that this young scientist, because he was a nobody at the time, although 3.5 decades later he went on to win the neuroscience equivalent of the Nobel, At the time, he was just a lowly assistant professor. And so they put him on the second floor of a building on the very edge of campus. But fortunately for this story and our sake, that building was the marine biology lab. And every day as he was walking into his work, one would imagine, he had to cross through this lab filled with fish tanks. And inspiration sparked. And he decided that what he should do is take a lot of the mazes that he'd been working with and put them into a water-based maze. And as a result, the olfactory cues would no longer be an issue. So he grabs a technician, they build this large tank over one weekend, they start testing it, and it works out beautifully. And basically what he does, it's so simple. He's got this pool, maybe very similar to what you would have splashed about in a very hot summer day when you were 4 or 5 years old. And he pops the animal down into it. And in the same way as if I popped you into a water pool, you'd be comfortable for a while, but you'd be motivated to get out. Well, rats are similar. They're quite good swimmers, but they don't want to swim in there forever. So they start swimming around. And as they're swimming, you can see in the top right quadrant, I've got this sort of dashed platform. Well, that platform is just under the surface of the water, so they can't see it as they're swimming. But as they're moving around, they bump into it by chance, and they get out. And they look, and around the room are situated different kinds of cues. And they see those cues, and they use them to orient the location themselves with the location of that escape platform. And in very short order, in successive trials, what you find is the amount of time that the animals require to find that platform, that is their escape latency, drops off remarkably, right down to a floor. Right? And in effect, you can see the ecological transferability of this, because when you leave here tonight, you're going to go somewhere, and you're going to get to that spot because you're going to leave this room and use three-dimensional cues to navigate through space back to the bus stop or residence or wherever the case might be. Okay? Good? All right. So with the water maze intact, we have a tool in order to assess how it is diet is affecting spatial memory performance. Right? The sort of last thing that's sort of most important to keep in mind is this task also critically requires a region of brain known as the hippocampus. And the hippocampus is important not only for memory formation almost in general, but in particular memories that have a spatial or episodic component. So let's take a look at the data. Okay, so we'll just kind of walk through the graph. So in the top right, you can see a graph, and on the y-axis is escape latency. So that's the proxy measure that I mentioned a moment ago for learning. Along the x-axis, you can see the training day. So typically what would happen is animals would go into the pool several times each day for a number of days. And as I mentioned a moment ago, over those successive training days, the amount of time that the animals require to escape goes down to a floor effect. Now what you can see here is there's no difference between the control diet animals and the high-fat diet animals. Okay? The bottom right graph is a set of data from where we play a bit of a cruel joke on the animals, you might say. So after they've been trained to locate the escape platform in that quadrant, we give them 24 hours, we put them back in the pool, but then we take the escape platform out. And the idea is that they, of course, will use the cues, go to that quadrant, and assume that it's there, and concentrate their search strategy in that spot. And the amount of time that they spend searching in that quadrant is what we take as an index of the strength of their memory. And think of yourselves. Think of your smartphone. So you're going to go home, you're going to pop it somewhere in your apartment or in your residence. That's where you do it. Put it down every day. And imagine if you got up tomorrow morning and it wasn't there. Well, you wouldn't automatically start searching in the bathroom, would you? You'd search in and around the area. You'd concentrate your search effort where you remember it being. So you'd look maybe under the table or in the drawers and so on. Similarly, the rats are remembering that the position of the platform is at this spot, and so they concentrate their search efforts there. Okay? So this is called a probe test. What you can see when we take a look at the data in the bottom right is when we assess the strength of spatial memories, there's no diet-related effect. Okay? So you're probably thinking, well, these are negative data, right? Well, negative data can be very, very informative, right? So it's critically important that we present them. And although what we were able to see is that high-fat diet causes metabolic shifts in adult female animals, it does that without impairing hippocampal-dependent learning and memory. And this is in contrast to some of the other data that we and others have gathered. So this is kind of the break point for the first part of the story. So what I'll do is just summarize a couple of things. So what we and others have found is that energy-dense diets can interfere with hippocampal-dependent memory, as measured by things like the Morris-Water maze. Right? So we found, for instance, that a high-sugar diet can interfere with spatial memory of this sort in male rats. We found that operant conditioning, you might remember this from psychology class, can be affected by high fat in male mice. However, there's a bit of a nuanced story here because the impairments seem to be sex-dependent in the sense that sometimes what we observe is that high-fat diets impair spatial memory in male rats, but not in female rats. Right? So in the interest of time, I haven't showed you this evening some of the other data that we've collected where indeed there is an impairment in male rats. Okay? As well, we've sort of tried to dive in to understand a little bit about what could be underlying some of these effects. And again, we don't have enough time this evening to go into those, but some of our preliminary data suggest that part of the explanation rests with shifts in the ability of the brain to handle insulin, and also shifts in terms of production of triglycerides as a result of all of that excess energy, and then the movement of those triglycerides into brain, and then actions on areas of brain that are responsible for governing learning and memory. Right? So for instance, several years ago we did some work where triglycerides were directly injected into the brains of animals, and as a result they were impaired on their ability to perform this sort of task. Okay? But again, just to sum up, it's really important when these kinds of experiments are done to first look at the sort of diet that the animals are on. You know, what is the composition of the diet? How is it being fed? What sorts of animals are being fed? Are they male? Are they female? What's the species? What's the strain? It's critically important to be very careful in terms of measuring the effects. What are the outcome measures? Both in terms of overall physiology, but then also in terms of behavior in biochemistry. In other words, this is a very complex field where it's very challenging to make definitive statements in a very broad sort of way. Although if we could distill what's out there, we might say that these energy-dense diets can affect metabolism and brain function and behavior under some conditions. Good? Okay. Well, what I have done to this point is just introduce to you some of the models that we use and some of the outcome measures that we take advantage of. What I really want to do now, though, is shift direction to the next set of experiments where we try to get at this question of, does maternal environment influence the way the brain develops? Right? So you might imagine that we would take advantage of these models, female animals, feed the diets, impregnate the animals, and then look at the offspring, right? So you can probably very quickly see where the experimental design is going. So this is very kind of complicated, but a moment ago I just kind of distilled the basic steps along the pathway. So we started with those same female rats, placed them on the diet that we just discussed a moment ago for about 10 weeks. Right? At the end of that, after we convinced ourselves again that this was a sufficient intervention to lead to a shift in their physiology, we then bred the animals over the course of a week. We then allowed for rodent pregnancy to go on for 3 weeks. So in case some of you had a burning desire to know how long a typical rat pregnancy lasts, it's 21 days. Right? This is a remarkable aspect of rats. Female rats can be pregnant within 24 hours of giving birth to a litter. And a typical female rodent can have a reproductive lifespan of about a full year. And a typical rodent litter can be 12 to 16 pups. So you see very, very quickly that in short order, one female rodent can give rise to dozens and dozens and dozens of offspring, which is one reason why they're a highly successful species. In fact, there are probably 2 or 3 times as many rats and mice in the city of Waterloo as there are people. But you don't see them, at least not typically, because we're blind to them. But if you sit outside this evening and you just be very quiet and watch, you'll start to see them come out. Go to your street during garbage night and watch. In any event, so about 3 weeks, they give birth. They then nurse for 3 more weeks. At the end of that point, they're weaned. Okay? And then the animals go on to just typical diets and then are assessed using the water maze at different points in the lifespan. Right? So keep in mind that we would have had a group of animals on these control diets and a group of animals on these high-fat diets, 45% of calories from saturated fat, during that 10-week period, during the breeding, gestation, and lactation points, okay? But that the offspring would have been placed onto the control diets. So really what we're focused on is effects of energy-dense diets during the very earliest parts of the lifespan. Good? So we've got male and female pups, and we assess them at 2 points in the lifespan. Now, before we jump into looking at the behavioral data, I want to just take a step back and look at a couple of other characteristics. One, the effects of high-fat diets on reproductive physiology, and 2, the effects of those diets on offspring metabolic physiology. Good? All right. So let's jump into some of those data. So take a look at the bottom left. And what you'll see in the bottom left are 2 different types of data. First, the size of the litter, and second, the sex ratios. I'll give you a moment or two just to digest those data. So what you can see by looking at these data is that there's a little bit of a difference in terms of the typical litter size between female rats on the control diet and female rats on the high-fat diet. And if we take a look at the next 2 bars, the sex ratio, we see that that difference is explained as a result of there being fewer male pups and more female pups. This was kind of an exciting finding for us because it is grounded in evolutionary theory. So in the early 1970s, there were a set of evolutionary biologists who put forward this notion that if the maternal environment shifts in such a way as to suggest that there's adversity, there's stressors, there's nutritional adversity, these kinds of things, sex ratios will also shift, and female offspring will be preferred. The thinking being, again, in an evolutionary context, that female offspring will have a heightened probability of themselves going on to breed and pass on genes to the next generation. So it makes good evolutionary sense for sex ratios to shift. And indeed, if you take a look at the practice of agriculture, people in the farming sector have known this for a long period of time, that sex ratios in domesticated farm animals can actually be modified simply by shifting maternal diets. So how exactly that takes place is quite unclear. We have some sense, as I mentioned a moment ago, from the Trivers-Willard theory of the broader sort of distal evolutionary context as to why that might take place, but how that translates down to the cellular physiology of the animals is very much an open-ended question. All right, so one thing to keep in mind. If we shift over to the other graph in the bottom right, what I'm illustrating here is just a bit of metabolic physiology of those offspring animals. And indeed, we looked at a whole list, more than a dozen different characteristics, to try to understand how maternal high-fat diet might be affecting offspring physiology. And these data are relatively representative of what we found. And that is a pattern of virtually no effect as a result of the diet. Okay? So interesting, because the diets were able to affect maternal physiology, but weren't affecting offspring physiology. And these results are notable for two reasons. One, they're somewhat inconsistent with the pattern that is typically seen in the literature, because if you take a look at the literature where people have fed modified diets, energy-dense diets of various sorts to female rats during the course of their pregnancy, what they will often find is some shifts in metabolic physiology in the offspring. What we found right across the board is virtually no differences. So it's notable for that reason. Although, as I mentioned a moment ago, people use all different kinds of diets, all different kinds of outcome measures, and so on, so there's a lot of technical variability. The other reason, though, why it's particularly important is that what we were truly interested in doing was going on to assess the behavior of these animals. And if it had been the case that there were shifts in their metabolic physiology, it might have made the interpretation of any behavioral differences that we went on to observe more challenging, because it would be hard to tease apart what might have been a direct developmental effect from what perhaps could have been downstream of a problem in terms of the animal's ability to handle glucose, for instance. That makes sense? Okay. So again, just a bit of a side note on the effects of the maternal high-fat diet, both on reproductive physiology and then on the metabolic physiology of the offspring. With those things set aside, though, what we can do is now jump into the main stage, if you will, and take a look at whether or not there were differences in spatial learning and memory in animals, in offspring, as a function of the maternal diet. Okay? And I've built it up enough, so I'm going to show you the data. Okay? And again, what I want you to do is just take a moment or two and digest these graphs. Now, we've looked at similar graphs a moment ago. Right? Along the y-axis, we have our learning measure. This is latency to reach the platform. Along the x-axis, we have training days. In each of those graphs, we've got a gray bar, offspring of mums who were on that high-fat diet, and a black bar, mums who were on the control diet. And what do you see? Are they the same?
No males are affected more.
Yes. So there's an effective diet, but there's an effective diet in a sex-specific manner. The male offspring had greater latency. In other words, they had more difficulty in acquiring the task, in forming those spatial memories. Right? Now, interestingly, if you think back to the schematic I showed in the last slide, we assessed spatial learning and memory at 2 points in the lifespan. This is the point right during the adolescent period. And again, another bit of trivia for you. If you've ever wondered how old a teenage rat is, right? A teenage rat is about 35 to 55 days of age. Right? So we assessed these animals during the adolescent period. When we returned and assessed these animals 2 months later, when they would have been young adults, we found that there was no sex difference. So what the data are suggesting is that maternal diet can cause an effect in a sex-specific manner, but that effect seems to self-correct in some way as the animals age. So this is interesting from a practical and from a physiological perspective. Right? From a physiological perspective, it's telling us that the diet is doing something. And from a practical perspective, it tells us that whenever we're measuring these kinds of potential changes, we need to be very intentional and very careful with when the outcome measures are collected. Because imagine if we hadn't assessed at this point in the lifespan and had simply decided to make the assessment when the animals were young adults. We would have seen no effect. Right? So again, sampling point, sampling frequency, these are very important technical considerations. Okay. So far, so good? Okay, wonderful. Now, if you're kind of looking at these data and you think, well, there's a sex-based difference, that's kind of interesting, right? Not entirely unexpected, because if we take a look again at male and female physiology, we see differences. if we take a look at neuronal circuitry and anatomical architecture between men and women, there are differences. More importantly, though, if we take a look at how men and women or male and female animals respond to different kinds of energy-dense diets, at the cognitive level, we also see differences. Right? So, for instance, other groups have shown that if you give male and female animals different kinds of energy-dense diets, you see differences in the induced gene expression in hippocampus and other areas of cortex. Or you see differences in neuronal firing patterns in various circuits within the brain. As well, if you take a look at humans, right, if you go to the Framingham Heart Study, one of the largest longitudinal studies looking at heart health and metabolic health more broadly, what you see is that men and women over the lifespan seem to show different cognitive effects related to differences in obesity and weight gain. And indeed, it's very, very practical in the sense that you could do functional imaging studies, and you could show men and women different kinds of pictures of highly palatable, desirable, energy-dense foods, and observe that different parts of brain light up and become active. Right? So all of these data, the animal level, the human level, illustrate that there are differences in male and female brain, and in particular differences in the way male and female brain is affected by food and by diet-induced illness. As a result, in the context of all of those data, it's not surprising that we've observed something like this. It's also not surprising that the sex that seems to be most sensitive are men, or male animals, because a host of research has indicated that male brain seems to be a little bit more sensitive or perhaps less resistant to different kinds of insults, right? Both diet-induced illness, but also hypoxic injury and so on. So with all of that discussed, what we can do is summarize the second section of the discussion. In other words, maternal obesity can alter breeding characteristics. It doesn't seem to alter offspring metabolism, but it does seem to affect offspring learning and memory. So, to this point, what we've discussed is what's often typical in the field. In other words, we've done something during early development that has led to an undesired outcome. Right? And if you take a look at the history of reproductive physiology, you see this pattern of people introducing toxicants, introducing modified diets, introducing models and interventions that are intended to cause some kind of undesirable outcome. Right? Well, what we wanted to do was shift focus a little bit and ask the question, well, can we intervene in some way that might be beneficial? Right? Instead of always looking at things that might be harmful, could we do something that's beneficial? Right? And if you think for a moment, you'll probably come up with a very easy intervention that lots of evidence would seem to suggest is beneficial. And of course, what I'm thinking about is exercise. How is it that exercise affects brain? Okay. Well, we and others have started to explore that question, but again, before we can understand how exercise applied to a pregnant animal affects offspring brain, we first need to understand how exercise affects the fully formed adult brain. Because in doing that, we can then discover candidates, things to look for in the offspring brain. That make sense? So what I want to do is now round out the evening by showing you some of the work that we've done in trying to understand how exercise affects the adult brain. So I'm not sure if the video is going to play. Take a moment and see. And no.
No, it doesn't like that, I'm sorry.
Okay, no worries. So what you can see in the bottom left is a rodent, and the little strip it's running on is a treadmill. And it's sort of colored red because we always run these experiments when it's nighttime for the animals, because keep in mind that their circadian pattern is different from ours. As nocturnal animals, they're typically most active during the nighttime or the dark phase of their daily cycle. Right? If we were to do these experiments during the light phase of the cycle, it would be kind of like me waking you up at 3 o'clock in the morning and putting you on a treadmill. Chances are your performance would be affected because it's not the optimal time of the day. So we run these experiments during the dark phase, and of course, as humans, we can't see very well in the dark. And so we have to put the red light on. Right? So the animal goes on, it's on this treadmill. We can adjust the speed of that treadmill to increase the exercise intensity. And across the top, you can see an outline of a schematic illustrating how we gradually train the animals to run on the treadmill and then over time increase the intensity. Yes?
But does that apply to, like, when you wake up in the morning trying to do like cognitive, like studying or working or something like that?
It's a little bit different waking up first thing in the morning versus waking up partway through a sleep cycle. Yeah. But it's a good question. Yeah. So on the bottom right, what you can see is a measure of VO2 max, and some of you, maybe if you're in kinesiology, will appreciate this measure. So it's basically a tool that allows one to measure how the body is using oxygen. And of course, as we're more physically active, we're going to use more oxygen. Right? So we can actually run these experiments by enclosing the animal in the treadmill inside a closed metabolic chamber so that we measure O2 going in, oxygen going in, and CO2, carbon dioxide, being produced. And using these and a host of other measures, we can get some sense as to whether or not the animal is working at maximal intensity. And that's what we see in the bottom graph with GXT. Right? So this is a graded exercise test to exhaustion. So we increase the incline of the treadmill and the speed of the treadmill until the animal is working at maximal aerobic capacity. And with that in mind, we can then titrate back and get a sense as to how to set the treadmill characteristics to have the animal operating at a moderate capacity. Make sense? So the reason why we wanted to do this was so that we can measure a graded intervention. So either sedentary animals, animals operating at maximal aerobic capacity, or those at an intermediate point. Right? So we had groups of animals. They went into these different exercise conditions. We then sacrificed the animals, took out different parts of the brain, and asked the very simple question, can we observe some kind of basic biochemical change? So if you've done a bit of biochemistry, you know that one of the major signaling mediators is phosphorylation. In other words, the ability of specialized enzymes to attach phosphate groups to proteins. And as a result of that phosphate addition, there's a shift in the structure of the proteins, a change perhaps in how it connects with other proteins, where it's shuttled within the cell, and so on. So phosphorylation is critically important in terms of mediating molecular interactions within the cell, and it's quite even more greatly important within the context of neurons. So we thought that this was a very important place to begin looking. So we asked the question, if we take a look across tissues in brain of animals that have been on these different exercise conditions, can we see shifts in phosphorylation? So pictured here is just a set of data from a standard Western blot. So we would have taken out tissues from the animals, we would have ground up those tissues, separated out proteins based on their relative molecular size, and then used specialized antibodies to allow us to measure the amount of phosphorylation at different points. And what you can very clearly see along the x-axis is that if we look at different molecular weights, we see all of those proteins seeing a shift in phosphorylation status as a function of exercise. Right? So with this, what we had in hand was a tool to allow us to determine that we could actually measure a biochemical change in brain tissue tied in with pattern of exercise. And again, I'm sort of outlining this carefully because if you're going to answer the question, does this intervention have some kind of effect, you need to very clearly identify the outcome measure you're going to use. Okay? So that's actually where we're at right now. Right? So we've developed this exercise model, and And we've shown that we can put animals through different kinds of exercise to different levels. And we've shown that we can match those different exercise levels with this neurophysiological outcome measure. Right? You can see very clearly now where we're going next. So what we would like to do is take female animals, put them through this training regimen or something similar to it, perhaps at different points in pregnancy, and then start to observe offspring to see whether or not some of the benefits of exercise that are seen typically in adult animals could shift the developmental physiology of their offspring. In other words, instead of simply looking at interventions that might cause a decline in development or some kind of undesirable change, we want to see whether or not we might be able to identify an intervention that could have some sort of good outcome. And of course, it doesn't take much thought now to then do the next experiment, which is where we have a high-fat diet animal, an undesirable outcome, and we intervene by providing the opportunity for exercise to see whether or not it might be counterbalanced. And of course, what we would want to do is make those assessments at a number of levels. At the tissue level as illustrated here, and then of course at the behavioral level as illustrated in some of the earlier slides. So with all of that done, what I want to do is just thank the people who actually did the work, thank the agencies who provided the funding for the work, and last but not least, thank you for your patience.